This is First Look SoCal 2020. Matthew Bresnahan is our moderator, and he is going to introduce our judging panel for the next session of Life Science Startups. Take it away, Matt.
Great. Yeah, thank you so much, Darren. As a reminder, same format as the last session we had, 3-minute company presentations followed by 9 minutes of Q&A with the judges. And as a reminder, to the extent anybody attending would like to ask a question, feel free to insert that into the Q&A box, and be happy to get to those questions in the course of things. So with that, we have a great panel of judges, actually a lot of friendly faces I see. So what I'll do, I'll just list the names and then allow each of you to introduce yourself. So we have Nancy Hong from RiverVest. Good to see you, Nancy. Zach Myers from my firm, Wilson Sonsini. JJ Rika from Pismo Ventures. Suresh Pai from LumaMed Solutions, and Paul Grant, who is the executive chairman of Cardia Bio. So Nancy, if you'd like to introduce yourself, maybe talk a little bit about RiverVest and what your areas of interest are.
Sure thing. Hi, everybody. This is Nancy Hong, dialing in from San Diego today. That's where I'm based. We have another partner down here, Niall O'Donnell. And headquarters for RiverVest is actually St. Louis, Missouri. The firm has been going for 20 years. We have a focus on life science, so medical devices and drug therapeutics. We like to be early stage investors. A lot of times we're the first institutional money in. We do also found companies, and we're always investing to have a board seat to help these companies grow. And so we're investing out of our current fund, which is a $185 million fund, probably do 10 core positions. So think of our bite size as often between 5, 10, 15 million, and then appropriately reserved. In terms of areas of focus, we do a lot of oncology, we do a lot of orphan disease. On the medical device side, we're doing a lot of cardiovascular and larger indications where the few remaining acquirers are interested. So that's a bit about us. I have a PhD as a background, love immunology, love oncology, and really looking forward to today.
Excellent. Well, thank you so much, Nancy, for joining us. Zach?
Yeah, hi. So my name is Zach Myers. I am one of Matt's corporate partners at Wilson Sonsini, focused exclusively on life sciences and technology. And I'm on the corporate side, so helping companies raise capital, incorporate, and grow their business over time. So anything that relates to that lifespan, from day one through your early Series C rounds to Series A rounds to ultimately going public and getting sold. My practice is almost exclusively life sciences with a little bit of additional tech. My background, I was a CPA before becoming an attorney, so really help to work companies, help companies grow and raise capital. So that's what I do, and love to work with early stage companies, and I'm really excited to see all of the new companies today.
Excellent. Thanks, Zach, and thanks for taking the time to join us. JJ?
Hello. Thank you for having me. My name is JJ Risha. I am with Pismo Ventures. Pismo Ventures is a venture studio. We work with early stage startups as well. We're agnostic as far as industry is concerned, from technology to healthcare to life sciences and everything in between. We are also we're building a fund this year as well, so we'd be able to invest in our portfolio companies. And as a venture studio, we basically provide, in addition to capital, human capital as well, if the companies also require that. I'm happy to be here. I'll do a quick plug-in. We're doing a venture plan competition, which is a business plan competition as well, which really the applications end on the 27th of September, which is in a couple of weeks. So hoping some of the current presenters will be able to join us.
Excellent. Thank you, JJ.
Thank you.
Suresh?
Yeah, hi. Thanks for having me. My name is Suresh Pai. I'm the principal at LumaMed Solutions. We're kind of a cross-section of an accelerator, an incubator, and I like to describe it as a medical device development studio. My background goes back in time about 25 years. Medical devices is our focus, mechanical devices and biomaterials. over the course of time, mostly spent a lot of my efforts and experiences on vascular devices, interventional cardiology, and peripheral vascular setting. But over the course of different startup companies that I worked with, we started broadening our horizons and worked on things in otolaryngology and laparoscopic surgery. And starting in around 2008, 2009, when the capital crisis hit, we ended up a couple of like-minded founders and myself formed entities under LlamaMed Solutions that kind of focused on fast to market 510K mostly, and some PMA based technologies that we can take from whiteboard all the way to commercial markets and oftentimes liquidity with very, very lean capital. So we've actually started a number of companies we've taken all the way to exit with mostly non-institutional funding model, and now we've sort of institutionalized that and built some infrastructure in the San Francisco Bay Area of about 12,000 square feet with clean rooms and an overarching ISO certified quality system where we do organic product development and we do the types of things I just described, as well as doing partnering, helping startup companies that maybe can lean into some of our expertise where we can participate in the upside with them.
Great. Well, thank you so much for joining us, Suresh. Paul Grant with us? Might be on mute. It's the tagline. Okay, well, maybe we don't have Paul, so hopefully Paul can join us. Great group of judges. Thank you all for your time, valuable time. I'm confident that the feedback and questions you all provide will be incredibly helpful for these groups. So with that, why don't we start with the first company, which is AquaTeX Medical.
Okay,
Yep.
All right, let me go ahead. So imagine yourself sitting in the emergency room of a hospital because one of your family member had a stroke. And doctor shows up and says, your loved one has a bleeding in the brain, and they need to block the abnormal vessels to prevent the next bleedings. So today there's a new technology called the neurointervention. Instead of opening up the skull and trying to reach the site, it can be treated using catheters. So the catheter can be inserted from the groin, advance all the way up to the neck, and go into the tiny vessels in the brain. And sometimes they inject a kind of glue to seal off those abnormal vessels. It's called the liquid embolic material. There are two FDA-approved liquid embolic material in the market. One of them is called NVCA, which is essentially a super glue. The problem of this material is that if a surgeon missed the timing of removing the catheter, it can get trapped in the brain and it can cause massive bleeding. So FDA issued a safety alert. The newer version of this glue is called Onyx. It has a toxic organic solvent. However, it has much better handlings. Although the FDA issued a safety alert in 2012 after receiving the complication of 100 patients, and half of them had a catheter trapped in the brain, and the 9 patients died. So the company also had to pay $18 million to settle the lawsuits from the Department of Justice. So there is a safety issue related to this treatment method. However, the number of the procedure is dramatically increasing in the past decades. The reason why is because the neuroendovascular treatment, the need for this treatment is growing. And also the market size of the liquid embolic material is estimated to around $100 million per year. So our solution from AquaTeX Medical is a new material called Aqua Embolic System. Aqua Embolic System is a liquid material. It can be injected from the microcatheter, and it becomes solid once exposed to the calcium ions in the blood. So it never catches the catheter. It's because it's made of the elastic hydrogel, and its main composition is there's no toxic materials included, totally biocompatible. So for the proof of concept, we've been evaluating this material using animal models, and now we're working on the 55th version of the prototype, which is very much close to the quality of the MVP. My name is Ichiro Yuki from the Department of the Neurosurgery, UC Irvine Medical Center. So we have a strong team, the 2 neurointerventionalists and 1 vascular neurosurgeon with more than 20 years of experience. And the material is designed and continuously modified by the outstanding polymer scientists from Japan. So essentially the 7 years of the research and development has been supported by basically the government-based fundings. And now we have 2 intellectual property from the 2 academic institutions. So now our next goal is to continue our preclinical evaluation using animal models and to get the GLP study done and have this IDE approval from the FDA so that we can go for the clinical trials. So we will be seeking for a seed funding from the angel investors and the venture capital sometime next year, probably. And we are looking for entrepreneur partners who can join our team. So thank you very much. And we're looking for the people who can join us to improve the quality of the stroke treatment, the safer and better treatment. Thank you so much.
Thank you so much, Ichiro, for a very good presentation.
Yes.
I'll open it up to the judges. Would anybody like to go first?
Yeah, Dr. Yuki, thank you for your presentation.
Yes.
Can you help us understand like what kind of the stages of the development are for you from where you sit today to, you know, reaching the commercial markets? You know, what kind of capital do you need and what are the kinds of things you think you need to accomplish to get there?
Yes, thank you very much for the question. So we are I would say, very early stage of the early stage in terms of the fundings. So as I mentioned before, we've been doing this research and development based on the government-based fundings, and we recently launched the company just a month ago. So we are trying to get the another government funding such as SDTR. The next cycle, we're preparing for it. And one more thing that we need to do before going to the GLP study is we want to make sure the long-term result of the large animal experiment. So we've done the small animals, the rabbit studies for the survival models worked, and we did the swine studies with the acute model, it worked, but we just wanted to have longer term efficacy and also the biocompatibility, so to make sure that it works for the as a preclinical evaluation. And yes, that is our stage right now.
So you feel that some of that non-dilutive funding will drive some level of value so that when you take money from preferred investors, you'll have some of that animal testing completed, for example, or are you looking to raise money ahead of that?
It really depending on, how much funding we can get from it, but I would say another $100,000 to $200,000 in having additional 6 to 9 large animal models, and if you can complete it, and we're pretty much ready for the GLP study.
Dr. Yuki, can you tell us a little bit about the competitive landscape in your space?
Yes, that's exactly absolutely a relevant question. So as I mentioned, there are 2 liquid embolic material you can use for the cerebrovascular disease at this moment, and both of them has limitations. So first of all, the liquid embolic material, those materials are now only indicated for the treatment of the AVM resections only, arteriovenous malformation. It's a cerebrovascular malformation. But people are using for the different uses for such the vascular malformation, vascular-rich tumor embolizations, other vascular malformations, and even the outside of the neuro, the body interventionalists are using this material for GI bleeding, so tumor embolizations. There are many different the applications. So at first, probably our first step is to create the material equivalent to the currently available material, and that's the first the market. And then, because it's biologically not material and it doesn't catch the catheter, the safer material, we can probably expand the specification of the material even further. That is our sort of step-by-step approach we're thinking about.
Are you aware of any competitors in development kind of in the pipeline?
Oh yes, there are so many similar, but I don't think any of them are reaching the pretty much ready for the commercialization. So there are similar projects going on from different academic institutions and using hydrogel type polymers. Yes.
How much do you need in order to get to commercialization?
I would say $1 to $2 million to get the IDE approval, and a final commercialization somewhere around $9 to $10 million. We estimate.
Okay, and the length of time, do you think? You've been at it for 7 years. How long do you think it'll take?
Shortest 5 years and 7 years, maybe 10 years. Yes, it really depends on how it progresses, I assume.
Okay, thank you.
Yes.
Dr. Yuki, have you met with the FDA yet?
We started just having the conversation with the FDA. Yes.
To discuss your regulatory path?
Exactly, sir. Exactly.
Okay.
And you mentioned you guys would go after the target indication that is already approved as a starting point because that would be the easiest for you, or did you have something else in mind?
Yes, it's absolutely relevant question. So this material can be used for, from our point of view, can be used for a wide variety of, the indications, using the body interventions and neuro interventions. But as a neurointerventionalist, and we are literally facing the problem right now, we want to tackle this cerebrovascular disease first. So we probably want to go for the same path that Onyx and NBCA and other liquid embolic material went through, which is the category of the neuroendovascular, the arteriovenous malformations and so forth. Yes.
What's the size of the target market and what would you expect to capture?
Yes, currently we're still working on this market analysis, the detailed market analysis. Currently our the number we have approximately is included in this market in the presentation. It's around $100 million per year, but it's it's only for the liquid embolic material, and also the particles can be used for the tumor embolization and so forth. But if you can expand if you can expand the indication further, it can be a $200-300 million per market. Yes.
Got it. And then the sales right now, I don't know if these are publicly disclosed for the product from Medtronic and the other J&J product. They're around 100 million, and that's how you were getting that?
Yes, it's a little bit tricky to get those detailed informations, but based on the publicized information, that those are the numbers we guessed.
Great. We did get a question from an attendee. Question is this approach indicated only for aneurysms?
Again, so there are many different types of the cerebrovascular disease. One of them is aneurysm. However, from technical point of view, the aneurysm treatment using liquid embolic material is a little bit tricky. So AVMs, dural arteriovenous fistula, so-called the dural AVF, those are the two main target first, and we may be able to use it for the aneurysm as well. Yes, so that is going to be the third goal. Yes.
So more than aneurysms, though. There's other indications as well. It sounds like, yes, including aneurysm. Great. Okay. Any final questions from anyone on the panel? Okay. Well, Dr. Yuki, great job. Thank you so much for your time.
Thank you so much. Thank you very much.
Thank you. So let's move on to our next group, which is BioJam Technologies.
Thank you everyone for being here. I'm Dr. Liu. I'm a robotic colorectal surgeon at UC San Diego. Today I want you to step in my shoes as we talk about a dilemma that faces modern surgeons like myself. And that's how to perform surgery safely by making as few incisions as possible. This is how surgery was and still is done. Open surgery is effective, but it's very gruesome and very difficult to recover from. Minimally invasive surgery is the principle of long instruments through small incisions, and patients like it a lot better. But the human, as you know, is a complex jigsaw puzzle with delicate organs that need to be moved around to show me where I'm supposed to operate. Without the retractor tools in open surgery, your organs would just be in the way, making it impossible to see anything below the surface organs. The best that modern medicine has to offer surgeons today is some archaic-looking fly swatter device that has numerous drawbacks. But mostly, it requires an additional incision to be made on the patient just so I can have an assistant hold on to it and push the organ out of the way during the entire case. But don't just take my word for it. These are common headaches among colleagues in modern surgery. The market to solve this is a global one, with annual markets of over $600 million in the US alone in 2016. Plus, more incisions means more complications, such as hernias. Eliminating even a single incision in half the laparoscopic cases has the potential to save healthcare $2 to $3 billion a year in hernia repairs. But most importantly, less incisions means the patients will recover better with less complications and better cosmesis. At BioJam, we came up with a soft robotics retractor that solves all the surgeon's needs with almost zero footprint on the patient's body. That means no additional incisions, fits the contour of the patient's organs, and functions independently. Here's some images of how the device works in a live pig surgery. The area of operation below the yellow circle is completely covered by the liver. Our device is introduced here into the pig abdomen through an existing trocar incision. The retractor then easily sneaks under the liver and lifts it out the way to expose the gastroesophageal junction so that the surgeon here can perform a gastric bypass. Although this example involves only moving the liver, its principle translates to all laparoscopic and robotic surgery. Soft robotics is a technology that has not really been used in surgery. To date, we've tested this device in several live pig models as well as a pilot study in cadaver, all with positive feedback. Our team is composed of both surgeons and experts in soft robotics, giving us the technical know-how and the rapid feedback from clinicians. We have robust partnerships with the clinical and research arena, which has allowed us for rapid prototyping and early animal testing. With your investments, we'll be supporting a unique and novel technologies that surgeons desperately need. So help us, your surgeons, to get the tools to help you.
Thank you. I just mentioned, great job, Dr. Liu, and turn over to the panelists, to whoever would like to ask the first question.
Can you provide some color on the regulatory pathway for the product and what it's going to take to get there from here?
Yeah, good question. So originally our plan is to go for an exemption pathway because this fits under surgical retractors tools as general purpose retractors. But if in the event that FDA comes back with, that it cannot be exempt, then it will undergo a 510K pathway. There are several similar devices in the sense that they are intra-abdominal retractors, such as the one I demonstrated, the Ethicon device with the endo paddle, that this would be a predicated model for our device.
What's the price point for the existing device, and what's the price point for yours? Or what is it?
The price point for the current market and what's our projected price?
For the current retractor that's being used versus your device.
Yeah, a lot of these companies actually do private contracts with the companies, and because of how much they sell, the price varies a little bit. But just from looking at what's available on the websites and what internal information I could find out, each retractor costs anywhere from $300 to $600.
And are they reused, or can they be cleaned and reused, or are they single use only?
That's a very good question. So this is actually two components. I didn't get a chance to talk about the details of technology because of the 3-minute limitation. But there's a reusable component where it's the pump control for the soft robotics, and that part we're planning to lease to the institutions or the surgeons or whoever may be using them. And there's a disposable component, and that's the retractor itself. And we went for disposable simply because it was easier to make a disposable a clean retractor than to try to deal with the reprocessing of a retractor of this complexity.
So you mentioned the market for the minimally invasive retractors is about, what was it, 600 million? Was that right?
Million for all retractors, yes.
So what the current competition that exists for sort of in the space you guys are in, how much of a dent has been made in that market? Is it significant yet, or is this kind of a new area in terms of doing retraction using these devices?
It's completely new. I've never seen anything like it. Yeah. Actually, looking at even the IP space, soft robotics is not used for this at all. And the most that people have come up with in terms of calling it soft robotics is actually rigid robotics in connection. So like, for example, the endoscope, that's technically, they're calling it soft robotics, but that's not really soft. That's just a series of rigid robotics. But using true soft robotics, actuation chambers, jamming technology, it's not been done.
Mm-hmm.
Dr. Liu, can you help us visualize what a potential exit might look like for a venture capital investor?
I think so. Good question. So, originally when we started this on this journey, because people kept asking me, because they know that I was a surgeon who was also interested in engineering. They say, well, make me a device that does this, make me a device that does this. And this is a trouble that I face this challenge all the time. I'm trying to do my best for the patients, but there's just no tools available. And so in terms of exit strategies, we saw it as either one, we could independently produce this and then make that independent company that produces the retractors, or we could partner with, say, Ethicon, who already sells endo retractors, or go into the robotic surgery space. We've had the fortunate happenstance of sharing lab space with both Verb Surgical and Ethicon within our laboratory itself. So we have been in communication with them as both possible partnerships for development, as well as further future exit strategies.
Can you give us an idea of what the size of the device, and is this patentable or not?
Yeah, so there is a patent submitted. We've gotten back some minor revisions. It's being submitted for final patenting right now. The size of the device, it's actually very variable. We picked one that's, in this particular one that I demonstrated, I think was about 4 centimeters by about 12 centimeters. And that's just measuring fitting to the abdomen of the pig models that we were using. The size is actually fairly arbitrary. We can make different sizes, but the beauty of it that because it's soft robotics, it will just fit, it will bend and fit in different ways so that it could be technically used as a universal size. But the real size limitation actually is the trocar size, because we're using an existing trocar by not making additional incisions. So a 12 millimeter trocar is the biggest that we can fit it through typically. And we can do a 15, of course, but then you have to be talking about doing a surgery that requires a 15 millimeter trocar.
That makes sense. So on the patent, what are your claims?
The claims are that we use soft robotics to provide a device to, I believe it was to displace and move organs for surgery.
Thank you.
Can you help us understand, like, commercially, like, what's the distinction, differentiates you guys from the competition? I know there's some companies that are already commercial stage with different approaches, right? So what's going to make folks go to you for your product instead of those that are already setting up sort of almost a beachhead for sales already, right? You're going to have to differentiate yourself nicely from that, I take it.
Yeah. So a couple things. Number one, I think the IP itself really stands alone in that it is not something anyone's using in the past. Soft robotics as a whole field is very new. A lot of it is being used in the aerospace and aeronautical region, first time using it in healthcare. In terms of other specialties is that I think, whereas everyone is trying to invent a new thing that uses some novel space, the actual design of the retractor is very intuitive to surgeons. It models off a malleable retractor that we use in open surgery. And because of that, it's very easy to teach a surgeon why this translates to exactly what they were doing in open surgery. And we developed hundreds of years of literature on how to do surgery using those type of retractors. So I think this will really make it both translatable and easily adaptable.
Great. And we have a couple, actually several questions that have come in from the attendees. One question was that, Dr. Liu, you mentioned substantial cost savings in avoiding hernia repair. How often is this repair surgery needed in follow-up to a laparoscopic procedure?
It's a good question. I'll tell you that particular number of $2 to $3 billion I quoted, that does make the assumption that people actually get their hernia repaired. Some hernias are asymptomatic, and sometimes people just put off the repairing that hernia because they'd rather live with the pain than pay the several $1,000s for the repair. And so either way, you're solving people's problems of having discomfort from relating to hernias, and that's not relating also to the disfigurement. And hernias always grow in size. Maybe you can ignore it for 1 year, maybe you can ignore it in 2 years, but they always grow in size if you don't fix them. And so ultimately it will be a problem for those patients.
And a separate question from someone else, but related, are there any issues with trocar movement when the laparoscopic retractor is in place?
No, because the actual connector that connects the device to the external world is less than 2 millimeters in size and is a soft, malleable tube. So it in itself is not really, does not hinder laparoscopic motion when you introduce a typical, 5 millimeter laparoscopic instruments through a 10 millimeter trocar.
Great. Another question, what is the plan for human testing? I take it that depends on your regulatory strategy and
It does. It does. And
are you going to do other work too, even if it's not required, but for marketing purposes? Like, would you consider doing a clinical study, for example?
Yes.
Even if not required?
Yes. Well, I think the clinical study, a human clinical study aside, I think clinical study, if I was talk about using in live pig operations, doing like survival studies and looking at their outcomes, that's something definitely we want to do. It takes a lot more resources than we have right now, which is why we're reaching out to get partners and investors to kind of help us get to that stage. Of course, we also have to get the material from bio-inert to biocompatible, and that's one of the next steps that really takes a fair amount of money and beyond our current level of clinical expertise to bring to. Once that happens, we would like to we already started cadaveric testing, of course, with bio-inert materials, but once we have biocompatible materials, we'd love to do clinical trials in live humans. Ultimately, they're very difficult to done in the US. We're open to the idea of doing it in the US or other countries. One of my partners in this company, Dr. Horgan, he's led various clinical trials, both in South America as well as in Europe. And so we're hoping to use some of our partners with the previously established bridges to continue that route.
Great. Well, I think our time is up. We could probably have 3 or 4 more questions for you, Dr. Liu. Great presentation and really great engagement on Q&A. Best of luck to your company.
Thank you so much, everyone. Thanks for your time.
Thank you. All right. Thank you again, Dr. Liu. Let's move on to Amplifica.
Hi, everybody. You hear me well?
We can.
Okay, great. All right, so my name is Maxim Plikas, and today I'm presenting on behalf of Amplifica. It's an early stage company that's developing novel solution for hair loss. Common hair loss, also known as androgenic alopecia, some of you might be surprised, not only affect men, which is about 50 million in US, but also a lot of women, especially postmenopausal, approximately 30 million. And as you can imagine, a large number of these people are seeking all kinds of solutions for hair loss. So collectively, they spend about $3.5 billion in US on a variety of FDA approved and majority non-FDA approved products, with majority of those non-FDA products being like really not effective. But since this is so personal to many people, they go in at great length to try anything and everything. So it's projected that the hair loss market globally will surpass 12 billion by 2025. And the landscape looks the following. So there are only 2 FDA-approved products on the market. So those are the most effective are pills. They have to be taken daily. They have a side effect profile, and also compliance is a huge issue since it's daily. There's a number of FDA cleared but not approved what's called red laser devices. In my opinion, those are not effective, and this is based on the literature. So the market remains really tremendously attractive for a novel type of a biologic solution to come in and with the knowledge of hair biology to regrow the hair. So, and just kind of capturing that is the number of biologic-based solutions that are currently at various phases of clinical trial, they're in the middle. So where do we come in is with our solution that's really been handed to us by nature itself. And surprisingly, nobody ever looked at it before. So a lot of you have seen what's called hairy mole. It's a type of nevus, and the odd and unexplained feature of this nevus is that they grow really long hair in it. It's unsightly, especially very unsightly if it happens on the face in young children. They tend to be big. So clearly this is something that you would not want. But if you are a guy looking in the mirror, like this guy on the right, he's probably dreaming about having these hairs from his mole grow on his skull. So what we've done, we've studied nevi, We teased out the molecules that are enriched uniquely in the skin condition that by themselves can induce hair to grow without the need of a mole. This is just an example. So this is one of our leading candidate molecules delivered on the agarose beads into the mouse skin. It's on the right. So you can see a tuft of black hair growing around it, while in control, there's no effect. So
Maxim, I wanted to get you to, you might want to share your screen.
I'm not sharing my screen? That's unfortunate. Nope. Okay, you guys should have told me earlier. Okay, so anyhow, let's do it again. Okay, so.
There we go.
Okay, there we go. Okay.
Anyway, we'll give you a little more time, Maxim. Why don't you focus on, maybe the handful of slides that you think are most important for.
Yeah. So this is essentially demonstrating our solution. So on the left is an example of a hairy mole. It's in a small kid, but you can see that this is like a hairline. If that would be a hairline that many would kill for, this is like a teenage boy hairline that people really seek. So our solution is to imitate the effect of hairy nevus without having to have this unsightly condition by simply delivering into the skin, signaling molecules that are uniquely enriched in the nevus. So that's what we're doing. And this is, again, an example of this. So this is a mouse study where we've injected our leading candidate purified molecule into the skin on agarose beads. It very specifically and robustly induces hair growth, and you can appreciate the hairs around it. Okay, so our scientific team is myself, founder, faculty at UC Irvine, and William Rassman, who is really legendary in the field of hair transplantation. He is behind 50 plus patents and inventions, including robotic machines for hair transplantation. We've assembled a management and directors team, and they have substantial experience in biologics and had successful exits in that arena. We hold an exclusive worldwide license over our patent filed through UC Regents, and we have plans for additional IP filing. So this is our timeline. We at the end of 2020, and we're now racing to finalize our proof of concept studies in an animal model, a humanized animal model. So I think it's looking promising, so I'm quite optimistic about that. Starts 2021, we go in full speed in the first days of January for Series A fundraising that would allow us to fund the rest of the preclinical developments, pre-IND meeting, and eventually phase 1 filing. So with that, I'll end. Thank you.
Great. Thank you, Maxim. Turn it over to any of our panelists that would like to start first.
Maxim, how active has the hair loss, biotech scene been? Are there a lot of startups in this space? And
yeah, so you missed it. So let me do a screen share. It's my fault again. So let me do a screen share of just how active it is. I'm going to go, let me just go to that slide. Okay, this is what we're looking at. So on the left, the very few FDA-approved products, those were discovered accidentally, actually. Nobody at the time was developing hair loss products. These are drugs for Propecia. Propecia is a drug for benign prostate cancer. Side effect in clinical trial was like people regrow hair. Minoxidil is a failed drug for arrhythmias, but had also hair regrowth effect. So these are the only two things on the market that could be used for androgenic alopecia. The laser devices, it's gimmicks, in my opinion. So stuff here in the middle, I mean, those are the names of the companies, a variety of biologics that they're developing. But I think what really truly sets us apart is that our invention, so our idea is inspired by nature. So it's in a human. So a lot of these molecules here are taken out of the mouse studies, and there's actually a huge divide between the mouse and human hair biology. And time again, it's been shown in biotech that just having a great mouse idea doesn't mean that you're going to have a great solution for human hair loss. Many companies went bust over that. So what we're dealing with is a natural human condition that we're just simply imitating. It's very common. Small secret, I have a small mole in the back of my shoulder and it's growing hair. I'm actually not even cutting it. I'm planning to take a picture for the paper on it. So yeah, so I think that's what sets us apart. And I'm pretty confident that we teased out the top leading molecule in the mole.
And then can you describe a little bit the humanized animal
Humanized
models
mouse
that you're
model.
planning to
Yeah,
use?
there's some proprietary aspects to this, but essentially what we're doing, because like I told you, just testing things on the mouse hair and the fact that it grows is not enough. So hair follicle, although small, is highly complex in terms of its biology. It's not a static tissue, it's highly dynamic tissue, lots of cell division, differentiation events, stem cell decision events. So what we do, we graft human hair onto the mice, and we have a separate protocol where we can actually have them go into what we call rest phase. And then during the rest phase, we present them with our molecules versus some control molecules. We can actually see if our molecules can speed up entry into the growth phase to actually kickstart the new growth, and that's what we're seeing. So it's a human hair on a mouse kind of system, and it's the best preclinical model in this case, because there's really no other animal model other than, I would say, chimpanzee that could replicate true human hair biology. And experiments in chimpanzees at this point would be completely unethical for something like that. So yeah, that's what we're doing.
So we did have a question come in.
Yeah.
I think it's a stem cell, but I'll ask the question anyway. What is the composition of the drug treatment? Can it be made GMP?
Yeah. It's a biologic. It's a protein-based molecule, so it could be most certainly GMP produced, and we've looked into that. One of our management team members, in fact, had a successful exit in a bio company that developed exactly that. So we have internal expertise in GMP production, and that's the plan. That's the plan.
So it's a peptide.
Sorry, I can't speak in any detail.
Okay, yeah.
But yeah, it's in that kind of arena.
Okay, got it. And you could synthetically make it and scale it.
Oh yeah, absolutely. Yeah, we can make it in a purified form, buckets and buckets of it. So that would not be a problem. And we know like it's a definitive composition, which sort of sets us apart from some other companies, because there are some biologic-based companies for hair loss or regenerative therapies in general that rely on collecting like secretome of cultured cells. We actually have dozens, if not hundreds, of all kinds of proteins, and the composition is very undefined. So in our case, we have a very defined composition made out of purified molecules.
Great. Yeah, I know, going back to Nancy's question, there's been some companies in the space that have just had astronomical valuations.
Yeah. This is why we're kind of looking. So there's actually a lot of opportunity for a new drug that's effective that will come in. So in terms of kind of like our strategy, we're not even thinking to take it all the way through FDA approval. That would require huge capital. We actually hoping to have an exit in phase 1 at some point, even perhaps as early as pre-phase 1, as long as we can demonstrate, prove to people who understand the field that what we got is really working on humans. I think I'm pretty confident about that. So there are big pharma companies that are now essentially have moved from internal R&D development to buying out companies, and Allergan, which is just next door, recently made a couple acquisitions in the hair field, biologic stem cell based field. Almirall, which is a Spanish-based big player in the skin biologics field. So we talked to both Almirall and to Allergan. They're quite interested. They just want to see somewhat more developed product. So we're in that direction now, I feel.
Great. We did have a question. Actually, do we have a question from a panelist? Go ahead.
I was just going to ask, how much are you looking to raise in the Series A next year that you have on your timeline? Yeah, so we need about 15 million in Series A. That's what we need, and I think it's very reasonable. So,
and have you brought in any money on a convertible note or a safe yet to date?
Yeah. So what we're doing now, so the xenograft proof of concept studies that we're completing is based on friends and family funds that we've raised. That's about 600. So we still, half through that, so we're good to last. And so we have a number of investors that put in money,
and some of them are quite interested in participating in Series A. But what we kind of looking also is to bring one strategic, ideally, that could finance the whole thing rather than bits from several groups. So that's so we're evaluating that.
Great. And then let's do one last question from the audience. How does your product address the patient compliance issue? Does it require multiple doses?
Great question. Yes, yeah. So, and I was referring to that. So, where our product will work is by stimulating dormant hairs to grow. And it's a cycle. So hairs grow in a cycle. So it's almost like with the cell cycle. Biologists will understand if you can enable cell to pass the checkpoint. So they go through G1 to S, they will finish the S phase no matter what. So hairs kind of behave the same way. If you push them through checkpoint to go into the growth phase, they will execute the growth. Meaning that if a molecule is inducing this checkpoint pass, and that's what our molecule is doing, you only need to apply it every now and then. So the way we envision it, it will be kind of like Botox. So every 5 to 6 months, you go to your doctor's office to get reapplication of the drug. So compliance here would be much higher. Especially amongst women, because women, and believe it or not, care a lot more about hair loss than men. And they tend to be a lot more compliant. And
it's probably not half-life dependent because it'll be locally applied.
Yeah, it will not be systemic. It's most certainly a local application. So it's going to be either micro injection or even topical delivery. So we think for it could be possible to do topical delivery, or it will be like micro injection that's minimal invasive, it's non-painful. And in the field of biologic and delivery into the skin, there's actually a lot of development in terms of this user-friendly delivery.
Sure.
Yeah.
Great.
Thank you.
Well, excellent. Thank you so much, Maxim. Great presentation and good luck.
Thank you.
Let's keep it moving for the sake of the schedule here. Let's turn to SBG Therapeutics.
So yeah, my name is Zori Shapashik. I'm the founder of SBG Therapeutics. And at SBG Therapeutics, we develop drugs that block the androgen receptor to address unmet needs in dermatology. They focus on the orphan inflammatory skin disease, hidradenitis suppurativa. So current therapeutic options for these conditions are either oral, decades old, or used off-label. We've developed topical analogs of the oral anti-androgen spironolactone to address these conditions in a way that would disrupt current standard of care. So one of the greatest unmet needs in dermatology is the painful condition of the skin folds called hidradenitis suppurativa. It's largely incurable. In fact, surgery often doesn't work. It's known to be androgen or male hormone driven. So in women, HS is treated off-label today with relatively high oral doses of the blood pressure medication spironolactone due to its known off-target anti-androgen activity. Unfortunately, many women cannot take oral spironolactone due to pregnancy, cycle irregularities, breast tenderness, headaches, and other side effects, other problems. But doctors and patients have told us they want a topical anti-androgen to better treat HS and other hormonal skin diseases. There's really no currently approved topical. So the problem is that high doses of the blood pressure medication, spironolactone, cause a lot of side effects. And women want the efficacy of spironolactone, but without those side effects. Our solution is what we term the topical super spironolactone that we call SPG3. So spironolactone today, as I discussed, is used off-label in women primarily to treat 3 multi-billion-dollar skin conditions, including incident HS. Those include acne, hirsutism, and hair loss. Hundreds of thousands of prescriptions a year, in fact, are written today for oral spironolactone in dermatology. A safe and effective topical anti-androgen such as SPG3 has the potential to reach peak sales of more than $500 million per indication. So the androgen receptor is in fact a highly validated target in the skin. Our data has shown that SPG3 stays in the skin when applied topically, works for hours or even days after a single application. Our plan is to start clinical trials within 18 months of funding. Topically, SPG3 will supplant the use of oral spironolactone across the board in dermatology. We've developed SPG3 internally and filed broad IP coverage internationally. Our studies have shown that SPG3 is safe and effective in a 21-day topical hamster skin oil gland model, as evidenced by less red staining and smaller oil glands or sebaceous glands. In this model, androgens drive oil production. So our goal is to obtain clinical proof of concept as soon as possible with SPG3 to support an exit or expansion into additional indications within 3 years. Both HS and hirsutism, or abnormal hair growth in women, clinical trial designs are well established and non-invasive. We are exploring both options with partners and investors. To that end, we're seeking $4.7 million to take us from today to the end of phase 1. I've personally funded the company to date. Our key functions are covered by very highly experienced drug development team. In particular, Dr. Ken Washenik is a world-renowned hair growth expert and dermatologist. He's the head of the Bosley Hair Growth Clinic. For HS, we have real great interest from 2 leading clinicians from Duke and the University of Miami. So in summary, we're trying to develop a topical drug to treat 4 multi-billion-dollar hormonal skin issues. Our lead compound, SPG3, has been tested and patent protected. We're 18 months away from the clinic, and we're seeking $4.7 million to support an exit within 3 years. Thank you.
Thank you very much, Zori. I'm sure we have some questions or comments from the panelists. Anybody like to go first?
This is JJ. Can you tell us how much you've invested in the company so far?
Yes, we've invested around $500,000 to date.
Okay, and how long do you think, can you get away with less than 4.7 million to really get you somewhere, or you need it all at once?
No, I mean, the bulk of that money is to complete the IND enabling studies to get an IND, but obviously there are different different tranches of funding that we could use to get to that point. Some of the most expensive studies, involve, I can show you, there's a slide, in manufacturing of the product, the tox studies, and those are like the 3 largest areas of expense that we'd be looking to get into to get an IND. We're considering potentially looking at Australia for some tax benefits, particularly in clinical development, also in Canada. So we're really we're running this company virtually. I mean, my background is virtual drug development. I worked at Kythera Biopharmaceuticals. It was all done virtually. It was sold to Allergan for $2.1 billion. So definitely we see, we're really looking for leveraging and really taking a low-risk approach to drug development. But yes, there are definitely tranches that we could use to get to that goal, which is the end of phase 1.
Okay, thank you.
Sorry, I don't know if I missed it, but so is this a small molecule or is it a biologic?
In fact, no, this is a small molecule. So spironolactone itself is a small molecule. It's been around for 50, 60 years. It's a steroidal structure. We just made it, enhanced it, developed a new chemical entity, new IP around it, so composition of matter, use, and formulation. And what we've done is we've turned it into a PROTAC-like molecule. So now it's, instead of being it's a tripartite molecule, we have one part that interacts with the ligand, with our target, the androgen receptor, and we've linked it to another molecule which it basically helps destabilize the androgen receptor, leading to its clearance, degradation, and really sustained inhibition. So we can apply it once. We can see sustained inhibition for extended periods of time. We can wash it out for days. The protein is suppressed, and then the protein is turned over a couple days, gets regenerated, and protein is re-expressed, and the biological activity is returned. So that's the basic idea. Spironolactone today is an inhibitor of just the ligand binding, and so that's how it works on the androgen receptor. But we've enhanced it, and the other key is we made a topical where it stays in the skin. Spironolactone goes right through the skin, floods the body. That's not desirable. We want it to work in the skin and stay in the skin, so we can actually use it in women and then in men, because there's a huge market for this in men as well, particularly in hair and androgenetic alopecia, which today can't be used in men because it causes feminization. That's not really desirable, in part because the doses are so high. I mean, for blood pressure, you're treating with 20 milligrams a day. For dermatology, you're treating 200 milligrams a day. So it's quite a lot of spironolactone today orally. If we go the topical route, we can drop that by an order of magnitude or more, keep it in the skin, and really eliminate these issues and make it widely applicable to both men and women.
In the preclinical testing that you've done with it, it looks like it stays in the skin for 18 hours. Is that based on the preliminary data that you have so far? I know you mentioned that time frame earlier.
Right, so we've done both studies in skin cells to look at target engagement, and from those studies we've seen extended activity or inhibition of the target for hours to days at a time. The other aspect of this, we've done skin permeation studies where we take the molecule, apply it to human skin ex vivo, and then we measure where it goes. So we look at penetration. We see that it stays in the skin, the dermis, and the epidermis. Very little of it actually leaks through into our receptor fluid, which is like, you can imagine that being the systemic circulation. That's what we know about it. It forms a depot in the skin, basically. Very little of it leaks through the skin. And that's due to not just formulation, but primarily due to the composition of the matter itself. The modifications we've made to it make it resident in the skin. And like I said, spironolactone is not like that. Spironolactone, if we apply it to this model system, it'll go right through the skin.
Yeah, and follow-up question on that. So do you have a new chemical entity, or is this a previously approved drug as the chemical entity itself?
So our molecule is a new chemical entity called SPG3. It's based on spironolactone, which is an existing generic drug.
Analog. So
it's
so
an
you're going down
analog,
the
yes.
full B1 pathway.
This is it's a totally new chemical entity. Has to go through that entire route of IND filing and testing and approval. It's not a 505(b)(2). It's not. There's no clear shortcut, but we've de-risked it as much as we can by taking something that's active and making it better, using a novel, sort of biological mechanism of action, the PROTAC approach.
Got it. Yeah, I thought you might be doing the B2 path based on Kythera, Kybella. But yeah, that makes sense. And you plan to take it through phase 1 and then ideally partner it?
That's correct. A lot of parties, a lot of investors, a lot of strategics are interested in it. Again, we spoke into the same ones you just heard: Allergan, Almirall, Leo, etc. Some of them have interest with basically a tox package or just a lesser degree of certainty. Some of them want to see human data. But in the end, if you can generate good human data, your valuation, that is a major inflection point for the company. At that point, really, you're in a good position to exit or do a larger raise and move into other indications. Maybe just split off one of the indications and continue the medical versus the aesthetic. So a lot of opportunities if we can get to human data. We think the cost and the risk, the cost is relatively modest to get to that point based on, the strategy we've taken, low risk, sort of unmet medical needs / small studies that can generate results in a short period of time. So clinical trials, we're talking about in the order of, 8 to 12 weeks, that's the duration of the trial. And we're talking about something like 20 to 30 patients where we can generate the data that we need.
So those data demands that you think that the strategics would want to see to partner with you guys, that was that sub $5 million investment you think you need to get to that point where you're attractive to them to make a partnership?
So that would be not only we're attracted to them, but we have a greater valuation to them. There's interest, possibly at an earlier stage as well, but a lower valuation. So I think that, you're right. Basically, that's like the optimum, I think, for this product in this category. You can raise more, but then you can take it to another level. Of course, take on that risk, but and that may be an opportunity as well. So we'd like to share the risk. Get good data early, partner, exit part of it, and then continue maybe down the road with other indications, or maybe just exit entirely. And as you just heard, there have been a number of exits in the space, early stage or major investments from Allergan, particularly in hair. And others. There's my extended slide deck. There's a number of these that are in the upfront multi-million-dollar deals, subsequently much larger numbers. So really a lot of opportunities. They're multi-billion-dollar indications, obviously large markets, large opportunities. There recent approval in this space, sort of. So obviously a lot of activity is going. We're specifically avoiding the acne space initially because of the challenges with reimbursements, with competition and generics. But that's a huge market as well. So our strategy is focused on the unmet needs, sort of the less competitive indications from market perspective, but those with great opportunities. And that includes HS. There's nothing really approved topically. Nothing works essentially except surgery. There's a Humira is approved, and that's actually $1 billion a year product for AbbVie in this narrow indication. Reason for that is it requires high doses. You need to use 4 times the doses of Humira to treat HS that you would do to treat RA. So potentially we could help payers a lot by reducing that need for Humira. We can work with Humira, make it work better. Still a lot of opportunities there in a derm condition. Nothing topical and really nothing that works that's approved.
Great. Interesting opportunities, Ori. Any final questions from any of the panelists? We've asked a lot and heard a lot, which is great. Okay. lots of time. So thank you so much, Zori. Great presentation, and best of luck to the company.
Thank you. Great questions.
All right, next up, we have Syntr Health Technologies and Ahmed.
Thank you for having me. My name is Ahmed Zobi. I am the CEO of Syntr Health. We are reducing diabetic amputations in the aging population. This is a few highlights that we've met so far. We've developed the Syntr FPU 360, which is a medical device that is able to process adipose tissue at the point of care. We've been approved to start human clinical trials with 24 patients. We've completed our animal studies, raised well over $1 million in both federal and non-federal funding, and we're seeking a small bridge round or super seed round of $500,000 to get us through our major milestones. So I'm going to warn you about this unmet clinical need. It is very disturbing. This is an example of a grade 2 diabetic foot ulcer. This is what happens when you leave that grade 2 diabetic foot ulcer alone or can't even treat it or heal it. It turns into a grade 3 where you could see that there's already a hole in the foot. Normally infections lead to the bone, and this will lead to an amputation. And unfortunately, a good portion of diabetic patients today do receive an amputation of either a limb or appendages such as a toe on their diabetic foot ulcer. So what's our solution? We take a small fat sample from the patient's abdomen, process it in our system, which is patented, and re-inject it back into the patient's foot to aid in the healing process. Our price is competitive. For the system, it is a reusable system, so we're pricing it at $10,000, which is pretty low for medical devices in this space. And then for our consumables, it's priced at $1,000. There are Medicare reimbursement. Sorry?
Oh, go ahead.
There are, there are medical reimbursement codes that cover up to $2,700 from start to finish of the therapy. So we do fall under those codes. Eventually, we'll move on to get our own codes. And we do have various patents that have been approved and exclusive rights to those patents. Gross revenue projection by year 6, or about 4 years from today, is about $100 million. This is just treating 70 to 80,000 patients. And the diabetic foot ulcer market is valued at 9 to 13 billion. This is the team. We're a group of medical device experts and biomedical engineers, along with people in the space like Dane Shackelford, Naveen, who's a consultant, and Ron King, a clinical research officer. We also have a clinical trials team that's made of key opinion leaders in this space that are helping us through our clinical trials. Major timeline projections, we have early sales with plastic surgeons starting next year, post 510K clearance at $2 million a year. We're submitting our 510K class 1, class 2, hopefully in the next month. We have been approved for IRB, so the goal is to start the trials and then seek the funding in order to meet those expectations. Quick breakdown on the clinical trials is about 175 will be used to start 3 to 10, patents, filing, and approvals, regulatory to get broad indications for use, and then expansion uses, and then product development to get our device for clinical use, and then administrative, hiring some research scientists to have on staff in order to do some of the research here in our wet lab facility at ULP. And that is it. Thank you so much.
Any questions from any panelists? Anybody like to go first?
Can you tell us a little bit about the competitive landscape today?
So that's a very good question. A majority of the competitors in this space right now, and I don't think I have the slide back up. I think it got canceled, but I could just tell you it. They're more skin substitutes, artificial skin cells that are donated, grown out in a lab, cut up into little pieces, and then sent out to podiatrist offices, and that's what they apply. There are people that are trying to target the diabetic foot ulcer space with a prevention model. So this is more of a, hey, let's try to predict that it's going to happen in the first place, attack it with, you know, diabetic socks and shoes and stuff. But as a son of a diabetic mother, I could tell you right now that doesn't really work out. A lot of the patients really don't listen to their doctors when it comes to treatment pathways. So what we're targeting with our therapy is that it's basically, it takes the patient out of the equation so that you can attack the problem with their own fat tissue, regenerate the fat layer that's in the foot that is gone due to the diabetic foot ulcer, so that they're able to regenerate that wound and then move on with their lives.
We have a question from an attendee, Ahmed. Any interest in broader categories of wound, for example, trauma?
That's a very good question. We've started talking with the Department of Defense, and we've put in a couple of applications with them for trauma in the battlefield to aid in at least starting the healing process. Like, for example, if a soldier's on the battlefield, an IED goes off, they can at least target the wound with a fat injectable, whether it's prepped already, cryopreserved, whatever it might be, or processed at the point of care right there so that they can get to a level 4, level 5 facility in the future. But the main target right now is the diabetic foot ulcers with the plastic surgery arm due to the fact that we are partnered with several plastic surgeons in the area that have been helping us acquire the fat tissue necessary to do our research. So they're super interested in buying our technology for the aesthetics arm while we focus, and we know that the long haul is the diabetic foot ulcer arm.
When I was looking at your use of proceeds slide, you were talking about maybe product development and regulatory being about $150,000, which is exceptionally lean, as you can imagine, for medical devices. Can you help us understand that? Have you guys done a lot of the legwork already underneath it? Because you have really 2 submissions, or at least 2 data packages. I guess one of them is class 1, and you're saying the other one's class 2, correct?
Yes.
So can you help us understand that? Because that's a pretty small amount of money. It's impressive
to
pull it off.
So we've actually spent roughly 100 to maybe 150,000 thus far on our FDA package. We've passed all required testings. We're literally just 2 bench tests away from submitting our package. So the 50,000 is really just to be used as a slush fund in case the FDA comes back with us with questions. So we're ready. We're about to submit it. We have the funds necessary to get it through. And the $100,000 for the product dev, that's more on getting the device ready for our clinical trials. That's just basically everything is done. We just need to make the devices, manufacture them, assemble them, and then ship them off to our sites.
And is that done in-house then, or do you guys outsource that?
No, so the manufacturing is done through a partner in the Irvine area. They have everything ready. We have the parts already completed and done. It's just a matter of pulling the trigger on the purchase order and getting things ready and shipped out.
Great, thank you.
Yep, no worries, thank you.
It looks like you've had a good amount of friends and family investments so far. Has the management team invested in the business as well?
We are undergraduates that graduated from UCI. We had no money starting from UCI, so we have just basically put in all of our time and effort into this. Any competition winnings that we've generated over the last 4 years, we didn't take a cut out of it. We just put it right back into the business. So other than time, effort, and hard blood, sweat, and tears, no, we didn't put any of our own money because we don't have our own money. But we have, been working on getting federal funding. We have a couple of them in the works right now. So we're hoping to secure those by the beginning of next year.
A couple questions come in here.
Yep.
This one's shorter, so I'll defer to that one. Who are the potential acquirers?
So that's a very good question. I don't know if I could share my screen again. That would be awesome. Let's see. Oh, cool. I could. So we have a couple of them lined up. We are in talks with some of them. The biggest indication that they want to see for any potential product dev or funding is human clinical data. So the goal is to try to acquire some human data. They didn't say that they want 100 patient. They didn't say that they want 50. They say, hey, show us that it kind of works in humans so that we can at least start talking about some product dev funding or even venture arm funding. So 3M, AbbVie, Johnson & Johnson, Novo Nordisk are the major players in this field. The reason why AbbVie is that they acquired a company called LifeCell, which is a hand grinding fat processing device, mainly targeting aesthetics use, large volume fat grafting, not so much on the wound healing application side of things. So we spoke to 3M. They recently purchased Acelity for $4.4 billion in cash, plus I think like $2 billion in what's it called? Milestone payments. And Acelity sold LifeCell to Allergan. So right now 3M doesn't have a device to process fat tissue. So we're in talks with various corporations. So there's a huge potential for quick acquisition post-human clinical studies or even early feasibility studies. So where we don't even have to go down the full path. But the goal for Syntr right now, the team and everyone on board, is to take this thing as far as possible.
Great. And then you had another question come in here, and this may be down the road, but the question is, will you be going into any other markets aside from diabetic foot ulcers? And a follow-up to that is, do the adipose-derived cells show any efficacy for osteoarthritis?
We don't have any data for osteoarthritis, but there are a bunch of people obviously testing stem cells for osteoarthritis. Just a big, I want to make this clear. We are not using stem cells. We are not isolating stem cells. We are breaking down the fat tissue and injecting that fat tissue back into the patient. So there's, as you probably have heard, there's multiple people out there that are isolating cells, whether it's in a, chiropractic office or whatever it might be. Those isolating stem cells is considered a drug indication for use in the United States. Now, it might not be in South Korea or Japan, but in the US, it is a drug indication for use. So the way we fall under the 510K pathway is purely by keeping the structure of fat the same, just mincing it down so that you can easily re-inject it back into the patient's body. The other, let's see, what was his other question?
There's a two-part, but
will you be going into other markets?
Yeah.
Yeah, right now it's the aesthetics. We've heard a lot of praise from plastic surgeons around us that we've been working with, so they're super excited. And as I mentioned, we have a pre-sale of about $2 million off of just one of them. The goal is to close multiple of them in the next coming months.
Great. Just a few seconds left. Any final questions for Ahmed and Syntr? Okay, well, great job, Ahmed, and best of luck to your company.
Thank you so much, Matt. Have a good one, guys. Thank you.
All right, and yeah, just want to thank all the judges. Thank you guys so much for your time. Lots of great input for these companies, and really appreciate you all taking the time to provide that to them. So thank you to each of you.
Yeah, Nancy, Suresh, Zach, and JJ, thank you so much for participating today.